This Week in Virology, the podcast about viruses, the kind that make you sick. Hello, Madison. It's time for This Week in Virology. This is episode 193. Today is July 24th, 2012. I'm Vincent Racaniello, and you are listening to TWiV, the podcast all about viruses. Today we are recording at an amazing place. We're right on a lake in Madison, Wisconsin. We're at the annual meeting of the American Society for Virology. And we have a great crew for you here today to talk about viruses. Joining me, my old sidekick from the University of Florida at Gainesville, Rich Condit.
Howdy, Vincent. Hi, everybody.
How you doing, Rich? Having a good meeting?
Oh, this is terrific. This is the heart of virology right here. It's a great society and a great meeting, and terrific presentations. What a great place to do a TWiV, huh?
Perfect.
Wonderful.
I'm just checking the temperature here. We had pretty hot weather the last couple of days. Today it's cooled off. It's been raining. It's 22 degrees and rainy outside.
That's like 86 for those of you who don't think centigrade.
But I understand you need the water out here, right? It has been pretty dry all summer, so we need it. But this is an amazing venue we have here. For those of us listening, you can't see it. It's a room in the convention center right along the lake, and it's all glass behind us. This is just spectacular. Last year, Ann Palmenberg told us we were going to have a spectacular venue for TWiV, and she was absolutely right. So let me tell you about our guests today. We have two, and they are speakers at the meeting. They've already spoken, so they're totally relaxed and ready to chat. From the Department of Microbiology and Molecular Genetics at the University of Pittsburgh, Carolyn Coyne.
Hello, everyone.
Welcome to TWiV.
Thank you. I'm very excited.
All right, glad to have you. And in the Department of Molecular Genetics and Microbiology, University of Texas at Austin, Sara Sawyer.
Hi, everyone. It's really great to see so many people here. We've been going from 8:30 in the morning until 10 at night, and yet here you all are still up for more at lunchtime.
And meeting's not over yet. We have another day, roughly, right? So there's plenty more. I wanted to ask you, you're not wearing your cowboy boots?
Yeah. I think you're alluding to the comment I made when you asked me if I wanted to do this. I think I said something like, well, you have me shaking in my boots, but I guess we'll do that, no problem. And I'm from Texas, so of course you ran with that.
Yep, I was meaning to ask you the whole time. We also have a great audience here. This room is packed, even more than last year, and we can't do this without you. If the room were empty, we wouldn't be asked back. So thank you for coming. And I want to tell you that you can ask questions during this episode. I have a wireless mic here. If you want, I can give this to you, and you can do that. Or if you're shy, you can send a tweet via Twitter. Use the hashtag #ASV12. And I'm going to try and look at the Twitter stream here. I don't know if I have enough bandwidth to do all this, to talk and watch the recording and do the Twitter stream, but I'll try and get some questions from people here or people outside, so people can listen to the, well, they can't listen outside, but they can ask questions here in the convention center. So before we get into some science, I want to just have Sara and Carolyn tell us a little bit about themselves. It's always nice to hear where people came from and their training. So let's start with Carolyn.
Okay, I'll start. So I received my degree in biochemistry at Florida State University.
Ooh.
That's right. Go Seminoles.
Can I move?
And stayed in the ACC and went to the University of North Carolina at Chapel Hill. Got my PhD in pharmacology. Never worked with viruses. Worked on a project that was very much a cell biology project, I'd say, as a graduate student. but was always very interested. I worked in the airway, and I was always very interested kind of in these complex airway cells, how viruses infect. And so when I was looking for postdocs, I specifically sought out someone who was working on a virus that was known to infect at least the gut epithelium, Jeff Bergelson, who works on Coxsackie virus. And I did my postdoctoral work at the University of Pennsylvania, where I really sort of got into the world of virology. It took me a long time to sort of acknowledge that I had the skill to refer to myself as a virologist, but I think at this stage I probably do. And so after leaving Jeff's lab, I started my own lab at the University of Pittsburgh about 5.5 years ago. And that's where I am now.
So I'm going to steal the Rich Condit question.
What's that?
When you were haploid, where were you?
So I was actually born in Flint, Michigan.
And grew up in Florida.
And grew up in Florida.
Okay. How about you, Sara?
Well, I have an unusual path to arriving at this table right here. I have a bachelor's degree in chemical engineering. I never had a single biology class in college, and I went to work as a driller for the oil industry as my first career. So I worked on offshore oil rigs for a company called Amoco. Do you ever remember Amoco gas stations? Only people over like maybe 35 know what an Amoco gas station is. Amoco was bought out by BP, but I worked out on drilling rigs with those roustabouts who live out there for 30 days at a time. I took a helicopter to work, and I stayed out there for a few weeks at a time and did all kinds of crazy things on drilling rigs. And then I decided that maybe, actually what inspired me to go back to biology was partly running away from the oil industry, but also partly a high school teacher that I had in Olathe, Kansas, where I grew up. And I had this, and it's funny, one of my colleagues at the University of Texas at Austin, who's now a professor in my department, had this same high school teacher. This was a phenomenal person. And I remember doing an experiment in high school in my genetics class. I don't know how many of you had genetics in high school, but I did. And we did this experiment where we were given 3 chicks. And over the course of a semester, we injected one with testosterone, one with estrogen, and one with saline. And we did that. Can you imagine high school students running around with testosterone-filled syringes? Somehow my teacher got around getting permission for this experiment. Clearly, he's deceased now, so I don't think I'll get him in trouble. But it was just a fantastic class, and I just could never get it off my mind. And so after I worked for the oil industry for a bit, I went to Cornell University, where I got a PhD in genetics and development. I studied the replicative helicase that unwinds DNA during DNA replication in Saccharomyces cerevisiae. Didn't do any virology. I then got basically fell head over heels in love with evolutionary biology somewhere along the way, and I went to work for this phenomenal person, Harmeet Malik, in Seattle. Maybe some of you know him. I was his very first employee in his lab, and it was like pretty much the luckiest thing that's ever happened to me. And at that, I went to work with him on speciation, how organisms become reproductively incompatible with one another. And while I was at the Hutch, I met Michael Emmerman, who was just an equally important person in my life and in my career. And he introduced me to viruses and got me working on applying evolutionary ideas to virology. And so and now here I sit. Yeah.
So there's a theme here that echoes what Sandy Weller was saying at her talk, which is you can do lots of different things, and the path is not necessarily straightforward, right? And you can't plan it. Just do what you want to do.
Yeah.
How long were you on oil rigs?
Well, I worked, when I was a college student, I worked in the summers as an intern, so in all it was about 3 years. I was based out of New Orleans and flew out of the bayou down there, and the helicopter pilots are all ex-Vietnam vets, so those rides can be pretty interesting, getting out there.
Were you ever frightened working on the rig?
Well, you have to go through training before you begin, and they made me go to this class where they took me to an indoor swimming pool and put me in full work gear and work boots and strapped me into a mock helicopter and plunged me into the deep end of a pool, and I had to be able to get myself out before they'd let me go out to my first rig. So.
That's what I do to my students when they sign up.
So I just have this picture of you working on this rig, I don't know, covered with oil or whatever, and you're thinking about science, right? Was that the case?
Well, so I was an engineer. I wasn't actually throwing that bit in the ground, but I was. It was just it was an interesting view on the cost of the consumption that we all do, and just this entire industry that exists for the purpose of bringing us power. And I'm grateful for the experience. That's an unusual thing to get to see.
Do you use any of that experience day to day in the lab now?
Well, if you can live around roustabouts 24 hours a day, then graduate students are downright delightful.
So that's an amazing story. I don't think we've had anything like that, right? There's probably it's hard to compete with that one. Anyway, if you guys want a job in science, first go work on an oil rig, I guess. That's the moral. So I have a question for both Sara and Carolyn. You're both pretty new in your labs, less than 6 years, right? Both of you. So what a lot of people out here are getting ready to start their labs or soon, and what advice would you give them? They're going to have an empty lab, and they need to get people in that lab, and that's the situation you were in. We all were in, of course. So what advice would you give them to get people into that empty lab?
Well, I think enthusiasm is sort of the number one. I think enthusiasm is infectious. And so for me, when I started my lab, and I think junior faculty and can go about it differently, but I, for a very long time, up until the last year, really have felt like I'm my own postdoc to some extent. And so I work in the lab probably much more than I even should, sometimes near full time for years. I did that. And so I think what helped me recruit people to the lab was that I was in the lab all the time. I was enthusiastic, and especially for students, what's been very nice is when they come into my lab, I'm there all the time, and so I know what's going on. I know the experiments that are going on. I do the experiments myself as well, so I think that's allowed me to really supervise and guide people, I would say pretty well. But I think it's just enthusiasm. I think it's being enthusiastic. And the other thing is I always consider myself, if I take anyone into my lab, be it a postdoc or a graduate student, that I'm really their advocate. And so I feel very strongly about that if a student comes into my lab or a postdoc, I'm their advocate to help them do whatever it is they want to do later. If a postdoc comes and says, I might a postdoc actually do this, say, I don't really think I want to stay in academics. I don't think I want to stay in basic research. I want to teach. And so from the very beginning, I sort of looked at it like my job to help her get to that point. And so I think people generally respect that, and I think there are people that seek that out, especially people that don't necessarily want to stay in traditional, basic research. So,
do you think the subject or what you work on has any effect?
Absolutely. Yeah, no, I think absolutely. I think they're kind of buzz, and I think those buzzwords and buzz things kind of change over the years. But, for me, I've always sort of sold myself and considered myself possibly, somebody who applies, cell biology to the study of virology. And so I think that's appealing to a certain group of people that, want to do kind of combine virology and cell biology.
How about you, Sara?
Well, I think I don't know if I'm successful at this. Some of my trainees are here, so maybe we could ask them. But I try to center my lab around a philosophical question rather than specific experiments or even specific viruses. We tend to work on every virus under the sun, whatever we have to work on we want to really just take a scalpel and open the beast and see how evolution works. And viruses are such an amazing tool to study evolution because they evolve fast. They force the things they interact with to evolve more quickly. And so you just get this anybody who claims that you can't actually see evolution happen has never thought much about viruses, because you can see all kinds of amazing dynamics unfolding in this realm. And so as an evolutionary biologist, there's no better place for me to be. So I try to structure a lab that's very around this idea, and boy, we spend a lot of time sitting around debating ideas, and I just try to really keep my interaction with my employees centered around that, because I find that question is going to motivate them more than I as a person ever can. So that's, I guess, my piece of advice.
We have a quote on your website by Patrick Forterre.
Yeah.
It says, "The billion years war between cells and viruses has been and still is the major engine of life evolution." So that's what you're talking about here.
Yeah, I think the vast diversity of form and function and that you see on the planet is, I think pathogens have played a major role in driving all those different tentacles of change and evolution that have spread out and spawned over the planet.
So why viruses? Why not other pathogens like bacteria, fungi, parasites? Why aren't they as major a driver?
Well, I think they are. I mean, definitely, I think they are. Viruses change the most quickly, so the race gets run at a little faster pace, I think, when you put viruses into the mix.
So you could use your approaches to look at pressures put by those pathogens as well.
Yeah, right? Absolutely. I think so. I think when I said that we can't actually see evolution happening, I mean, we can even see human evolution happening, astonishingly. I'm sure you're all familiar with, the malaria story and sickle cell anemia and how these deficient hemoglobin alleles are at high frequency in areas of the world where malaria is at very high prevalence because these alleles protect against malaria. But in a homozygous state, they can also have various anemias and other problems. I mean, that's the human genome having been shaped by a pathogen in a very real and immediate way, and in a way that's still ongoing as well. So if we got rid of malaria, if the Gates campaign was successful, all of those sickle cell related alleles could millions of years from now be gone from the planet because there'd be no advantage anymore. So
viruses historically have had a place in biology because they're genetically simple, okay? So they're easier to understand, and it seems to me that applies in this case as well. When I think about the kind of stuff that you do, the ability to look at just a few genes as shaping the evolution of something seems to help. Is that true?
Yeah. Maybe I don't quite understand the question.
Well,
Sorry.
I mean, you can look at the virus attachment protein or a particular antiviral protein or something like that in a fairly simple genetic context. Seems it's more straightforward than looking at bacteria or fungi or something.
That's
like
right. There's just simpler. So it's easier to tease out the one-to-one relationships between the host and the virus. Right. I think. Yeah.
So everyone in this room probably heard your talk, but this recording, of course, is going to be heard by many, many thousands more people who didn't. So as sort of a backdrop for more discussion, could you pick one of your systems and tell us how you identify these arms races, a specific story? You can pick any of the ones that you like and just give us an overview of how you do this.
Well, if you envision every human gene as a slowly moving cog. It changes very slowly over time. Gene sequences are relatively stable because we're highly evolved forms. But you picture a virus as a tiny little cog spinning very quickly because they have sloppy polymerases and they change really quickly. If one of those fast spinning virus cogs engages with one of your slow spinning human gene cogs, it will speed up the rate of evolution of that human gene. And the idea is, that's just an analogy. The way it actually works is people that have randomly mutations in their genes that make them survive infection with bias get to reproduce the most. And so over time, those allelic forms become the most common and predominate, but then viruses will respond because they're so good at counter-evolving, and then that will create selection for some new form. And so really, I consider myself an evolutionary geneticist, where we try to go in and look at those variant forms of human or other mammalian alleles that have arisen because they have advantage in the face of infections and pathogens. And really it's just another way to do genetics. There's really nothing more profound to what we do than that. Instead of, I often think about, many people like to do mutagenesis in a lab, and it's really easy to make loss of function mutations like alanine scanning and things like that. It's harder to make gain of function mutations because you often don't know where to make those mutations and what amino acid to change it to. Well, by looking at solutions that different animals have come up with, you can reconstitute those mutations and almost always get gain of function situations because those are unique solutions that have come up to unique challenges in individual species. And so it's just this rich reservoir for ideas for mutants to make in the lab. And I think that's pretty much what it boils down to.
So you start it seems to me that you start by looking at evolution of, say, some human gene that you know encodes a protein that's going to undergo some sort of specific actual physical interaction with a virus gene. Is that right?
Yeah.
So, examples?
Yeah, so a really good example of where this kind of thing can play out is with an immunity protein and the virus that it detects. And specifically like the pattern recognition receptors and the restriction factors and viral sensors, there are different terms for these things, but the proteins that recognize patterns specific to pathogens. That's like battleground number one for these evolutionary races, because that's where small changes in the pathogen can make them all of a sudden able to, slip by like a ghost and not be detected by these systems. And so then the minute that happens, there's strong evolutionary pressure for some person or mammal or entity out there that has a form of that sensor that does detect that pathogen. All of a sudden, they get to survive with bias.
So you pick a known interaction, right? A virus with its receptor or an immune protein, and then ask, is there evidence for change with evolution? So you look at different species, basically,
Yeah.
right? So we need to have sequences, obviously, from all of that, and that wouldn't have been possible X number of years ago, right?
Yeah, that's really true. If you're going to use evolution to study a microbe, then you have to look in the species where there's been some interesting evolution with that microbe. So if you care about HIV, you wouldn't go looking for evolutionary change that's happened in rodents, for instance, because HIV came from primates. And primates are the organisms that have viruses very similar to HIV, simian immunodeficiency viruses and related other retroviruses. And so we always have to think about, where did this human pathogen we're interested in come from? And what's the sink of organisms out there that have already been playing many hands of poker with this thing for many millions of years, and how can we go ask them for some winning solutions, basically. So we're working on arenaviruses of the New World rodents, for instance. These are some emerging pathogens in humans, like Hunine virus and some other emerging pathogens. These are coming from rodents. So to ask that question with those viruses, we have to go to the rodent species.
So could you do this in an unbiased way and look at the whole proteome and ask which proteins evolve the fastest, and then try and find viruses that interact with them?
Yeah, and some of that has been done. One problem is that we don't have, even though this is the age of genome sequencing, and boy, we do have an awful lot of amazing genomic resources that we didn't before. We still only have, for instance, with primates, we only have 7 genomes that have been solved. And because we're humans and we love ourselves so much, we solve the genomes of all of our closest relatives, which isn't very useful from an evolutionary perspective. So more resources would definitely improve the power of these techniques, but already we've been trying to do things like that, and we'll only get more sophisticated and be able to make more insights when we have more resources.
So when you look at the whole gene, let's say the human genome, what fraction of proteins evolve the fastest? Is it half or less than half?
No, I think we're talking about a special, the cogs that are on the rapid rate that are turning sort of, I would say, exceptionally faster than the rest of the cogs in the human genome is about 5%. And these are really enriched for just a few categories of genes. That little group of genes is chock full of immunity genes. It's also got some genes in it that are involved in sensing of the environment. Olfactory receptors evolve really quickly because your olfactory receptors, as species spread out and get new territories and stuff, they have to discern new smells, and they have to discern poison from food. And so this is a really intense system of evolution in a lot of mammals, is the smell and sight system, because this is what keeps you alive in multiple ways, helps you find your food and helps you avoid the things that are going to kill you. So there's a lot of evolutionary pressure on those genes
but
as well.
those have nothing to do with pathogens,
Nope.
right?
Yeah, so you certainly pathogens aren't the only thing that can drive rapid evolution of genes. Yeah.
And are there surprises, genes that don't fall into immune categories or sensors that might be worth following up, right?
Yeah, so my lab has one of those. I sort of have a little obsession with this. We've found that these non-homologous end-joining proteins, this is a pathway in your cells for repairing double-strand breaks. And for some reason, I first found this when I was a postdoc with Harmeet, and we found it in yeast, and now we've shown it's true in primates and humans as well. For some reason, you have two pathways for repairing double-strand breaks, homologous recombination and non-homologous end-joining. Homologous recombination genes are nice and boring in the evolutionary sense, the way you'd think they should be, right? But these non-homologous end-joining genes are very weird. They're very rapidly evolving. And we think this might have to do with the fact that it's kind of crazy. You know, we think about, everyone has their definition of the immune system, but if you think about, for instance, if you think about adenovirus, the DNA repair machinery links the genome of adenoviruses up into huge concatenamers so that they can't do anything. They're basically incapable of replicating. So there's an example of your DNA repair machinery acting as an immune mechanism that's protecting your cells. So the definition of immunity is very malleable, and we think that these DNA repair this DNA repair pathway may be involved with sort of fighting free double-stranded DNA ends that are encountered in many viral life cycles.
So you said at your talk, our hypothesis is that DNA repair genes are being shaped by antagonism from retroviruses and retrotransposons. So is this what you're talking about?
So there are many interactions between viruses and the DNA repair pathway. Retroviruses are special because they have to integrate into the host chromosome, and that's really the only family of human viruses that's doing that. And so they have an integrase protein that makes a break for them to access the chromosome, but that integrase protein is incapable of sort of sealing the deal, so to speak, and fixing those ends at the ends of the integration. So the virus relies on cellular repair machinery to do that. And because this is not a homologous repair, it's a foreign piece of DNA that's just put itself in, it relies on non-homologous end joining, we believe. And so that's a vulnerability in the cell, this non-homologous system.
So can you see evolution in those functions that look like they're I mean, there's a huge history of retrovirus infections in vertebrates. Can you see an evolution, rapid evolution in those genes that reflects that experience?
I think we do. We haven't. So the idea here would be just like with the other types of immunity genes. You know, if we could take different forms of these primate non-homologous end joining genes or repair genes and show that they have unique specificities against viral targets, then that would be really sort of good evidence that they've become refined for whatever they need to become refined for in different species. And so we're working on that.
So you're not really a virologist, right? I'm wondering where this is going to go. Do you think of yourself as more of an evolutionary biologist?
I think of myself as someone who studies innate immunity. You know, I think of a virologist as somebody who tweaks the virus and figures out how to put the virus together and how it puts itself together and transcribes its genome and translates its proteins and makes mutations in the virus. You know, we're basically tweaking the human immune system and then using the virus as a readout to see if we made things better or worse, basically.
Okay, so if you're interested in innate immunity, virology is going to be on your table for a long time.
Yeah.
So I'm still puzzled by this DNA end joining. I thought DNA was DNA, right? You're saying it could be different in a pathogen and therefore the host can change to evade that specifically and not affect its own function?
Well, so I think the best example here is, so Matt Weitzman has done a lot of work on adenovirus. And we know that DNA repair proteins concatenate these adenovirus genomes into long strings that are dead-end products. But the virus makes antagonists of these DNA repair proteins. The virus fights back, like viruses always do. So the virus makes proteins that mislocalize these DNA repair proteins and get them out of the nucleus so the virus can do its business. And so I think there's a war there, just like so many other examples we have where viruses make proteins to antagonize real immunity proteins rather than DNA repair immunity proteins.
I'm looking at a tweet here from Welkin, who says, evolution is to virology and all of biology what Kepler's laws of planetary motion are to astrophysics. Good?
Yeah.
Oh, he's feeding you. That's
Nice,
what he's doing.
Wilkins.
He's always good for those choice.
He must have a
summaries.
grant up for review soon.
Nice. One other thing I wanted to ask you about, I think I picked this up reading your website, are the trim proteins, which you've worked on quite a bit. And something unusual is going on at that locus, right? And could you talk to us a little bit about that?
Yeah, so this is I was really excited to see at this meeting, TWiV sort of made a huge splash in the HIV field almost a decade ago now, when Joe Sadrowski's lab discovered it as a major restriction factor against HIV. And in fact, Welkin Johnson has done a lot of the important work on TWiV, as have many other people. But I was excited at this meeting. Almost every session I sat in on, even the workshops on specialty other viruses, I don't think I saw a single session where there wasn't a mention of a TRIM gene at some point. So I think it's important for us to all acknowledge there are 100 TRIM genes in the human genome. And I think I'm going to predict, I'll put some money on this, I'm going to predict this is a big family of pathogen sensors with unique specificities, and that this is going to be the next toll-like receptor family or something like that. So I think these are dedicated pathogen sensors, at least half of them. They're sort of one half that has a special domain at its C-terminal end called the sexily named B30.2 domain. But I think this part of the family are a family of pathogen sensors, and I think they've been uniquely tailored, as the immune system will do, to see different pathogens. And I think that's why every session I sit in on, I see a different TRIM rising to prominence as being sort of the protein that community has identified as a big antagonist for its virus. Now, you might be alluding to, my lab recently published a paper that showed, so you have to think to yourself, a lot of immunity genes exist in big gene families, the toll-like receptors, ApoBex, TRIMs. And obviously that's smart, because if you could have one immunity protein and it can only be evolved to recognize one kind of thing at a time, why not duplicate that to 100 so that you can have 100 defenses all lined up against different things? That seems like a good strategy. But how does that happen? How do you go from one gene to 100 genes that all look kind of the same? Well, I mean, for decades, we have in genetics focused on SNPs, single nucleotide differences between all of you in this room. But there's been a real revolution going on in population genetics. We now appreciate that all of you differ by big chunks of genetic material as well. And because these chunks often they're big enough to contain multiple genes, then we say that your copy number variable for those genes. So if I have an extra chunk over you and it bears 3 genes, then I have higher copy number of those genes than you do. So this is the field of CNVs, or copy number variation. And it turns out that, of course, the way you go from 1 gene to 100 genes is that one individual experiences a duplication of that gene through this copy number variation process. If that person survives with bias over other people, that will go to fixation, and then there may be another duplication. And that's how this slow process of this amazing immune system that we have, gets developed. And so we've found that about 30 of the 100 human TRIM, about 70 of the TRIM genes in the human genome, everyone in this room has. But about 30 of them, you're only lucky enough to have if you're a rare individual on this planet. There are 30 that are still in flux, and some people have them, they're still polymorphic, just like all the polymorphic SNPs you've ever heard about in your life. And so we've identified big clusters of these things, segmental duplications that are found only in Asians for the reason that they arose there, most likely, as random mutational events. And so it's important to think that, and I have to say, Welkin Johnson and some other folks that he works with did copy number variation studies in macaques, and lo and behold, if these TRIM genes aren't copy number variable in macaques as well. So in separate primate species, we have individuals having different numbers of TRIM genes. It's important to think about when you're doing live animal model systems and things that you've got individuals that aren't isogenic. They not only could have SNP differences from each other, they might have whole genes that the control animal doesn't have. So something to consider.
How long does it take for a beneficial polymorphism to propagate through the human population? rough idea?
Yeah, it all depends on the strength of selection. If there's strong selection, it happens faster. I think that I think the current estimate is that variation that exists amongst our genomes now all reflects evolutionary events that have occurred in about the last 10 to 100,000 years of human evolution. So things tend to sew themselves up in about that amount of time, meaning they either disappear or they go to fixation or some place in between. But yeah.
One thing that was sort of bothering me during your talk, you mentioned this idea. It's nothing
Carolyn needs to talk a little.
Yeah. This is my last question for you. It's bothering me because I'm inadequate at resolving the problem, not because it's an issue. So you mentioned this idea. So this is the opposite. A mutation in a human gene will occur that gives a pathogen an entry, that lets them in the door, and then you mentioned this idea of then it will refine its ability to utilize whatever it needs past the door, right? But so what I thought is that original mutation, why would it be maintained in people at all?
Absolutely. So that was a little bit of a contrived experiment. I'll just recap that for the podcast and people that didn't have every single slide of my talk memorized maybe out there. But so we did an experiment where we took a cell surface receptor for a virus called mouse mammary tumor virus, and we took a species that is not infected by this virus. And by making some key substitutions in its cell surface receptor, and by the way, we used evolution to guide our mutagenesis because we substituted amino acids into this receptor from the species that this virus does infect. When we did that, we made the receptor of this new species susceptible to infection by this virus. And so he's asking me, well, why would that ever happen in nature? Certainly animals aren't selected to be more vulnerable. Like if a mutation like that arose, there would be, no advantage to be more vulnerable. The point of that experiment was a little bit different. The point was to show that, have you guys ever thought about why we seem to get so many diseases from primates? And why certain types of mammals tend to give us more of the diseases that we get than others? A lot of in the evolution literature, a lot of people debate that the lower the genetic divide. This is kind of obvious, right? The lower the genetic divide between two species, the easier that the pathogens will hop. And we were trying to show that at these key amino acid positions that we had focused on through this evolutionary analysis, that it was really similarities at those positions that created the easy entry path for new viruses from one species to another. So it was I kind of had to brush over that in the talk a little bit. Yeah.
But it could also be that these changes are beneficial in some way for the host, right?
Absolutely.
And just randomly happen to help a virus get in or a pathogen.
And then it would be a scenario of weighing two costs against each other, which is the bigger problem for that species. Yeah.
All right, let's move on to Carolyn.
My turn.
Your turn. So when you were talking about placentas this morning, can we talk about placentas?
We can talk about the placenta.
You guys finish your
Who doesn't
lunch,
love
right?
a good placenta talk at lunch, I mean, honestly.
They're done eating.
I didn't show any pictures, by the way, this morning, and that was on purpose.
So I was just picturing our first son was born 18 years ago, and I remember that placenta being carted off in the tray.
Not good looking.
It was huge.
Huge.
So you must get tons of
cells. So
you get the whole thing?
The whole thing. It's actually, so the collaborator that I have at the Women's Hospital, Yoel Sadovsky, does most of our cell isolation, and they take the entire placenta, and depending on, because obviously the placenta size does vary actually from mother to mother, but generally speaking can get upwards of 60 million cells per isolation of this particular kind of placental trophoblast cell that we're working in, and so basically a placenta will give us enough cells that we can freeze them. So you can actually freeze them down, and you'll have a lot of cells.
60 million. So a bunch of T75s.
Absolutely, yeah. And these are the specific syncytio trophoblasts in particular. So these have been sort of selected out from the entire placenta.
Let's get the world clued
I know,
in
because
on
I didn't
this.
know either 4 years ago.
So,
yeah.
so the basic theme here is that you're looking at the placenta as a barrier to infection of the fetus, right?
It's how the project started actually, because I became pregnant, and I work in a lab with viruses. No, it's true. So what happened was I became pregnant, and actually the postdoc in my lab was pregnant at the same time, and we work with a lot of viruses. And so she and I looked at each other, and you know, if anyone in the room has had a baby, or certainly, you know, fathers who have pregnant women, you know, especially early on, you worry about everything. Everything that you eat, is this okay? Is this going to hurt the baby? And so, you know, we would be sitting in the tissue culture hood with, you know, Coxsackie virus and you know, these other viruses saying, well, I mean, is this okay? And that's actually what started this entire project 100%. And in fact, the first slide that I showed were actually 3D ultrasound pictures of my son. And I keep those in my talk because that's what inspired, you know, the entire project. And so we became interested in learning just about what was known about the placenta. And what was really somewhat shocking to me was how little was known about the placenta in terms of a barrier, in terms of especially viruses. And so that was really what started the entire project. And we were very, very fortunate that Pitt had recruited Yoel Sadovsky, my collaborator, about a year after I'd arrived. And he's a tremendously talented OBGYN who's a placental biologist. And so we were really fortunate.
So let me make sure I'm testing myself here.
Okay.
The cells that you have studied as sort of the primary focus of this study are in the epithelium of the portion of the placenta that's actually directly juxtaposed to the maternal tissue, right?
Yes.
So it's physically even the first line of defense, right?
100%. So they're a specialized epithelial cell type, so there are two layers of the placenta, cellular layers of the placenta. Placental cells are composed of trophoblasts, and so the outside layer are the syncytio trophoblasts, which are bathed directly in maternal blood, and then right below them are the cytotrophoblasts. And so absolutely the syncytio trophoblasts are the primary cell barrier because they're constantly bathed in maternal blood.
They sound really weird, like multinucleate?
Multinucleated, terminally differentiated, yeah.
And ciliated, right?
And
so
then
there's
completely
a lot of
ciliated,
surface.
they have junctions. I mean, so they really are, for someone who had worked with polarized epithelial cells for quite some time, completely different even from the cells that I normally have worked with.
So you get this big thing in your laboratory.
We get the cultured cells, so it's not nearly as ugly. We just go over
but
to
now
our
you got
lab.
But you must have seen the process.
No, of course, yeah.
Okay, so how do you get these cells out of that thing?
Well, so I personally have never gone through the process, but generally speaking, it's the kind of usual way, honestly, that you get that you do a trypsinization process and then you do a post-plating process to get rid of fibroblasts. I mean, it's honestly the same way that we used to culture primary airway cells. I mean, it's a very similar process.
So they have there's a differential affinity for the dishes once you plate the cells?
Yeah, well, so basically, you sort of can get rid of the fibroblasts by putting them in an uncoated matrix. The fibroblasts will attach much faster, and you can go through a series of those. And it's probably, I mean, Yoel's, if he's listening, is probably just like in the corner. It's probably a lot more complex than how I'm describing it. But in general, that's how it works.
And they just sit down. They remain polarized?
They do. So we can grow them for 72 hours in culture. And pretty much almost exactly at the 72-hour time point, they will die. So we keep them in culture. They can, generally speaking, depending on the experiment we'll have them on, you know, a collagen-coated support. But yes, they stay polarized, they stay differentiated, and how we actually know how good our placental preparation is we essentially do a pregnancy test. We measure HCG, and so that tells us whether our preparation is good or not.
Do these cells actually turn over pretty fast in the placenta as well?
Well, yeah, so throughout gestation, the cytotrophoblasts actually give rise to the syncytio trophoblasts, and so if there's any damage or the cytotrophoblasts are there to sort of provide the repair, so to speak.
Would it be a bad idea to immortalize these?
Well, there are a couple of cell lines that are generally more representative of cytotrophoblasts that exist, and you can actually use different agents to differentiate them into syncytio trophoblasts, but those agents can sometimes cause some off-target effects.
If you can do it with primary cells, man.
Well, and that's kind of what we've been really had.
Sounds like you have no shortage of them.
Never.
Right?
I mean, and quite frankly, before we met Yoel, my graduate student and I actually met with someone in the women's hospital, and so we were asking, well, how do we go about getting a placenta? And his answer was, bring a bucket. I mean, and that is 100% true. I won't say his name in case that's some sort of like IRB problem, but he truly did. He said, "You want a placenta? Go get a bucket." And it was really and so to generally to get placental cells, they're considered waste materials. They go in the trash unless someone asks to eat them or something. But they do.
You do need an IRB.
You do, and we have to follow obviously the guidelines for that.
So basically, you told us this morning that these cells are particularly resistant to virus
Absolutely.
infection. Roughly how many viruses have you looked at so far?
Well, so we've probably looked at, when I said in my talk this morning, it's not an exaggeration to say that we literally walk the halls of anyone at Pitt with like conditioned medium from these cells and say, okay, you have a virus, and I would say the same thing to everyone here, because it's very easy to test it because you can apply the conditioned medium to whatever permissive cell type you have. So we're in the process now of probably testing, at least 5 additional viruses. And after my talk was approached by a couple of other people that have some other really fantastic viruses to test. So
once again, for the people out there who didn't hear your talk, you have a monolayer of these things.
Yes.
You challenge them with virus, and then they don't infect, and then you can harvest the oh no, wait a minute.
No, so the conditioned medium experiments, so we know that if we infect these cells with virus, none of the viruses we've screened really replicate to any significant levels. But the observation that was sort of the jumping off point for our project was I was really into innate immune signaling and was thinking about type 1 interferons. And so my assumption was, well, perhaps the placenta just generates high amounts of type 1 interferons. And so the question we asked was whether conditioned medium from these cells could confer resistance. And so to do those experiments, which is why I say we have sort of a seemingly endless supply of the reagent, is all we do is culture the cells in their regular growth medium, and then 48 to 72 hours after being in culture, just remove the media, and then that becomes the conditioned medium that we then apply to permissive cells.
Okay, and that makes those cells less permissive.
And that confers resistance to viral infection as well.
So then the question becomes, what's in that stuff?
Oh, and that was a fun time. Those are some fun days. No, they weren't, because we had this effect. And it was like, how do you figure out what's in the media? What do you do to then go back and say, okay, what's in this condition? It could be anything. And so, there was the graduate student who's unfortunately not here in the audience, but can attest to that those are probably pretty stressful days for a second or third year graduate student, because we had this really sort of enticing effect of this antiviral phenotype, but really weren't sure how to approach it. And it was really sort of a light bulb went off one day as we were kind of talking about what could it be, and we thought, well, why don't we just subject it to a lot of really simple treatments, sonication, freeze-thaw, RNase, and let's just see if we can get a handle on this. And then, the key thing that led us to sort of the next step of the process was to see that sonication abolished the effect, as did freeze-thaw.
Where did that come from? I mean, I can think of all sorts of things I can do to try and trash something, but sonication.
Oh, really? So I thought of vesicles. I mean, maybe it's sort of, so I was thinking if there were any kind of vesicles. And of course, I was familiar with this concept of exosomes, so I was maybe biased in that sense. But I was thinking, well, if there are any kind of vesicles, or they're going to be destroyed by sonication, so let's try that. So I think, and then that was really, once we had that result, that was when we kind of were, I think that was really the launching point for the project, going somewhere.
So let me, before we move on, let me ask you a couple of viruses. Did you try polio, I assume?
We did, yeah.
Resistant?
Absolutely. Yeah, and that was shocking because, as I'm sure you know, and even for Coxsackie virus, it infects, most human cells that we have to, fairly efficiently.
The reason I ask is many years ago we had a transgenic model for polio, and we could show the virus able to cross the placenta. So, and do you think it might be different in mice?
Oh, I do, because eventually. So for people that either weren't at the talk or at the podcast, we've sort of identified this cluster of microRNAs that seems to be conferring this resistance, but that cluster is not present in mice. So that is a primate-specific microRNA cluster, and nobody. I mean, I'm sure people are probably thinking about it, and probably the evolutionary biologists in particular. But why they exist, why they're there, no one knows, obviously. But so we're limited in terms of animal systems, and I think in using animal models for specific, potentially for the placenta, because these microRNAs don't exist. So we've never looked in
There could be a different mechanism, right?
Absolutely, there could be a cluster of microRNAs or something very similar to the cluster that we see.
Are you going to look at it in mice?
We would love to. So we actually, so we're actually in the process of generating transgenic mice that express the entire cluster, with the hopes of being able to sort of move a little bit more in vivo.
So during your talk, Welkin also he tweeted a question. What about endogenous retroviruses, which presumably go in there to infect the fetus, right?
No, it's an interesting question, and so I will say too. So some of the data that I didn't show, just to avoid confusion, was that we have actually identified some viruses whose replication goes up in the presence of these microRNAs, and so it's possible it's just it's virus specific to some extent. I mean, and depending upon the cellular pathway that we see induced, I think it's very likely that we may see differential effects, and that's really why we walk the halls and sort of ask people to test, because that's what we want to see, to then tease out the mechanisms of that. Yeah.
So Welkin also brought up another good point.
He asked a lot of questions, that Welkin.
He said,
look at him in the corner.
you want to, hey Welkin, you want to say, no, I can do it for you? Yeah. I'm giving you credit. You can. He said, syncytian is thought to be the product of endogenous retrovirus, right? And so could it be that a retrovirus is involved in making placental cells resistant to viruses in general? Did I get that right? More or less.
I mean, honestly, at this stage, who knows? I mean, we've been really primarily studying the mechanism in the cells that we've exposed to these microRNAs. But I think the mechanisms to protect the placenta itself from infection are probably much more complicated than just these microRNAs. Because, again, from an evolutionary perspective, you don't want just one layer of defense. I think what's going to be there is multi-layers of a defense mechanism. And so, I think that there could be tons of different things involved in promoting defense.
So I have a question here. Sorry.
Rubella. So
yes.
After the talk, actually, someone approached me who works on rubella, which we were very excited to hear, because what we would like to do is take viruses that are known to be particularly adept at crossing the placenta, or at least infecting the fetus, and testing the effects of this conditioned medium in our microRNAs on those viruses. We have CMV data, which is sort of intriguing, but I would love to test rubella. So that's definitely on the list.
So we have a Twitter question from Coyne, who says, are the proteins responsible for placental barrier similar or related to blood-brain barrier proteins?
Well, I mean, they're junction proteins. I mean, so the placenta expresses the same junction proteins that any epithelial or endothelial layer would express. So they absolutely have junctions. We've never measured transplacental resistance, so I don't know if in terms of development of resistance, only because it's difficult to do that just with our culture system in 72 hours, and I don't know how they would actually do plated on transwells. But yes, their junctions look very similar to the junctions in any other polarized cell type.
So, go ahead.
So the entity in the conditioned medium turned out to be exosomes.
So it turned out to be exosomes, which we purified and could basically recapitulate. So if we just took this conditioned medium and purified the exosomes, we could mimic this induction of the antiviral effect. If the conditioned medium that was now depleted of exosomes, which we also deplete FBS exosomes as well, because FBS actually does contain exosomes. So that's sort of the background media that we're using. And that the media depleted of exosomes then loses that effect.
So for the uninitiated out there in TWiV land, and for me,
yes.
Okay, exosomes. It's a vesicle that comes off of cells, right? Yeah. Where do they show up in biology?
So
is a synaptic vesicle a kind of exosome?
I guess it could be viewed in possibly some sense of the way. So exosomes basically arrive in multivesicular endosomes within the cell, and their processing is really complex. And I think a lot of people are looking into that, how they bud, how they're released. Why they function, I suppose you could ask, is why do exosomes exist? And I think there's probably a lot of answers to that, but I definitely think that it's becoming accepted that exosomes are becoming realized as mediators of sort of communication between cells.
Okay, and in this particular case, it turns out the exosomes are delivering
microRNAs.
Okay.
And so we were very fortunate, because that was not something, maybe people that work on microRNAs would have seen the exosome result and thought, oh, okay, microRNAs. But, for us, it wasn't, quite that simple.
People who work on microRNAs see microRNAs everywhere.
I suppose, yes. But we were very fortunate because, the collaborator of mine, Yoel, had been working on this placental-specific cluster of microRNAs. And so Yoel was actually the one that when we had the exosome results, that I think it's microRNAs. And I, talked to a number of people during that year and thought, oh, there's just no way that it's going to be a microRNA. And he was saying, oh, there's this placental-specific cluster. And I just thought, no way are we that lucky. That never happens. There's no possible way. And, we would meet, and, Yoel and I meet every couple of weeks and sort of, had these brainstorming sessions. And he always came back to these microRNAs. And I was really a skeptic. I really was. And then it was, when we did the first experiment, and it was one of those moments that you have where you think like, oh my, I can't believe we were right. I mean, like, when does that happen? So we basically mapped that the microRNAs that were present in these exosomes that were conferring resistant are actually from this large cluster that is only expressed in primate placenta.
So that's the value of a good collaborator, right?
Well, absolutely. I mean, I
Different
think it's the
viewpoint.
perfect example, too, of, what we benefited from was sort of his clinical expertise and obviously his expertise in the placenta. And what we sort of brought to the table was a little bit more of the virology and host side of things as we started to delve into, the possible host pathways that could be restricting infection.
And so the microRNAs induce a response in the target cells that makes them resistant. And that
Yes.
response?
Well, we think the response, I hate to sort of, so I think one of the responses is definitely an induction of autophagy. And I will say, if anyone has worked with autophagy, or, I think everyone's probably dabbled in it to some extent, it was pretty much, we wrote a list of pathways that we thought we would look at. And, autophagy was basically last on our list. And it was because we hoped it was the one that it wouldn't be. Because it's, I think it's complex and it's complicated. And so, we went through what I think most virologists would probably go after first, which is, it's type 1 interferons, it's got to be, some upregulation of a pattern recognition receptor or just overall induction of type 1 interferon, and it was not. And we looked at NF-kappa B, and we looked at sort of cell death and apoptosis and necrosis and whether these pathways were being modulated at all. And it was really, it was actually to the credit of the graduate student in my lab who had always had sort of an interest in, Elizabeth Delorme Maxford, by the way, who had always had an interest in autophagy and really kept sort of fighting or at least, bringing up autophagy. And I would say, let's try this next, let's try that next. And then finally I thought, like, okay, let's look at autophagy. And, we are by no means autophagy experts, but, when we sat down at the EM to look at our first grids of these cells, I don't think it took an expert in autophagy to say, like, what is that? What is going on within the cytoplasm of these cells? And so that's really how we identified autophagy.
So the cells in the placenta that are making the exosomes to package and deliver these microRNAs, they must be intrinsically resistant because they're making the microRNAs to start.
Absolutely. So the cells that we use are resistant. Yeah.
Right. So they're conferring resistance to whoever's listening.
The target cells, yeah. And we, of course, that's obviously something that we'd like to know more about is what, especially during pregnancy, what are the maternal cell types that are being targeted by these exosomes? As I mentioned in the talk, which I find just fascinating, is that you can actually detect these microRNAs in the serum of pregnant women throughout pregnancy. They really peak in the third trimester, and then 24 hours post-delivery they disappear. And so we know that they're in maternal serum. We know that they're circulating. We don't know that they're in exosomes in that state, but we suspect that they are. So potentially these exosomes could be targeted to any cell types throughout the body, but.
And you don't know exactly what the targets of the microRNAs are.
Oh gosh, I wish we did.
And that's going to be a mess, right?
That's complex, yeah. And as anyone, of course, that knows about microRNAs. And so when we first started the project and we were looking at 46 microRNAs that could have, redundant targets, or we thought, and so that's probably the number one thing that we're working on currently, because I think once we know the target or targets, I think then we can work backwards and sort of map these pathways together, I think, a little bit better.
So you're going to be working on placenta for the rest of your life?
I don't know. We kind of hop around a little bit in terms of our model cell type. But I will say that, I think I think it's a fascinating organ, and I think, like I said, and I don't know if it's because I've had a child or I'm a woman, but I find it very interesting and something that I'd like to pursue, just to provide knowledge on the placenta. So we will likely stay working on the placenta.
You've got other stuff in your history. Is there other stuff still going on in the lab, or is everything?
Oh no, actually, I've only had one graduate student who's really primarily been focusing on this project over the years, and obviously a couple of people in our collaborators lab, of course. No, so generally my lab sort of dabbles in lots of things. I think the central core of the questions in our lab are centered around sort of these cellular barriers. So we study the polarized endothelium of the blood-brain barrier, the polarized epithelium in the gut, and so again, at least to us, these placental trophoblasts sort of represent just one additional barrier to study. And so, we study a variety of aspects of host pathogen interactions or host virus interactions in those cells.
I have a feeling that other people are going to pick up on the placenta, because just the basic biology involved is very interesting.
It's very interesting, and I think there's a real gap there. I mean, to the point that even like the NIAID is recognizing this, and there are RFAs that are coming out almost yearly, asking for proposals based upon pregnancy, and not necessarily the placenta specifically, but I think it's recognized as an area that could really use a little bit of more work.
Sara, do these microRNAs fall in the category of rapidly evolving sequences?
I haven't looked at them, but maybe we should collaborate.
Yeah. So we've gotten to do we have a question?
Have you tried inoculating your cell system with small numbers of already infected cells, not released virus, but already infected cells?
So taking the primary cells. Oh, can you? Oh, sorry. The
lymphocytes already infected with any herpes virus, and put them and see what happens.
So the question is, instead of directly infecting our primary placental cultures with virus, to take infected immune cells specifically, or just other infected cells? We have not. No, we have not.
I had a.
That's how most
Where is it?
viruses are delivered to the fetus.
Well, it depends on where they're. I mean, obviously, under a state, I think it depends on the viral system, actually, more so than. I mean, so I think for some viruses that could potentially be true, but I think for many others that's probably not true. So I think for some viral systems that's likely how it could occur. But I think that there is also just the ability of viruses that are present in the maternal blood that would then directly interact with these cells. I think could also and does also occur.
I have one question from Twitter from Rachel. Have there been studies looking at autophagy induction viral infection in vivo guinea pig model of gestation, for example? There's
actually a really fascinating study that was published a number of years ago in Nature, and I have blanked on the authors' names, that looking at the survival of neonates in, I think it was ATG7 deficient knockout animals. And those animals actually don't survive very well in the neonatal phase, because the animals actually require, I think, there to be induction of autophagy. And so I don't know of any studies in guinea pigs or other animal models, but certainly within the vast array of ATG knockout animals, it has been studied, not necessarily from the context of the placenta, but more in the context of neonatal survival. And I think that was their model.
Actually, it occurs to me that we left one thing out for people out in TWiV land, which is the autophagy is basically the cell's garbage disposal, right?
Oh yeah, absolutely.
And so presumably the induction of autophagy makes it so that when you infect those cells, the virus enters and gets in.
It absolutely does.
But the autophagy pathway then eats it.
Well, we think that's what happens. I mean, I think there's obviously debate right now in the field about how antiviral autophagy is and under what circumstances and conditions is autophagy antiviral. And I can certainly say in our system that we see robust induction of autophagy robust induction of an antiviral effect, and C, actually, at least for VSV, that incoming particles are actually shuttled into these autophagosome/autolysosome structures. And so, in our hands, I think we're fairly convinced that autophagy is playing a role. There could be other pathways that, of course, are also, being upregulated or altered. But I definitely think autophagy is.
Well, there are 47 microRNAs.
I know.
You got your work cut out for you.
I know, that's going to be tough.
All right, let's read a couple of emails. We get great feedback here on TWiV, and maybe our panelists can help us answer them. The first one I actually saved for this episode is from Angela, who writes, "Hello, TWiV. Welcome to the great state of Wisconsin. I hope you enjoy your stay. I suggest in your free time that you try some fresh cheese curds from a local creamer, and maybe some fried ones too. I'm sure that those from the area can let you know where to get the best curds. I have yet to try a cheese curd, maybe tonight." Next, thank you for all the hard work you do as a senior in high school.
All right.
Right?
Yeah.
We get a number of these. Who is passionately in love with viruses. TWiV never ceases to bring joy and an education regarding the world of viruses. It has also provided entertainment while sewing my prom dress. And Angela sent a photo of her very nice prom dress and her prom date, so I'm glad we inspired that. TWiVers aren't just lovers of science, but also creative. On a final note, a question. As being a senior, I'll be applying for college and undergraduate positions at college labs. I was wondering if you fine men have any suggestions for the whole process. Should I inform these scientists that even though I have had no opportunity to do lab experience, I have tried to become familiar with the world of virology through TWiV and reading books such as The Great Influenza? Thank you, Angela.
Yeah, I think it's always good to share your enthusiasm. Why not? I think if someone's going to judge you for being excited about something like that, it's probably not either someplace you want to go or someone you want to work with. So yeah, absolutely.
And I'd say,
Okay.
getting a research position early on is a good idea. And yeah, you just go talk to people.
Yeah, tell them you love viruses, you're inspired by these books you read, and TWiV is better. And they'll say, "What's TWiV?" And then you can introduce them to it. Let's do one more. This is from Dina, who writes, "Dear TWiV hosters with the mosters. To Vincent, to ring your recollection bells, we met in Dublin before the SGM meeting, before your panel with Wendy Barkley, my PhD supervisor, and Ron Fouché of ferret transmission fame. At the time, I promised to send you an email to request that you and the TWiV team discuss ways that scientists can increase serendipitous encounters with new ideas. Of course, there are many ways to learn about new techniques or cellular pathways that might relate to one's research, including reading papers and attending conferences. But I'm interested in the activities in between, the once-a-month kind of activities where you get to interact with other people on your level who are abnormally excited by the nitty-gritty details of molecular biology, virology, epidemiology, or immunology. What drove me to write today is because I was trawling through the literature trying to find a journal article that would entice enough people to attend our voluntary journal club and hopefully stimulate an interesting discussion. We do all right for a department of about 35 postdocs and PhD students. We provide pizza and drinks and usually keep about 10 participants entertained for an hour. Our journal club is run in the traditional way. There's a presenter who reads an original research article thoroughly, uses PowerPoint slides to present the figures as he or she explains them. We ask questions during the presentation and have a general discussion at the end. But there is a little voice inside me that says this could be better. Therefore, I would like to ask you, the TWiV hosts, and your listeners to share your experiences of journal clubs or other brainstorming discussion mixtures that you have tried and what you have found that makes that experience a little bit extra special. Thanks in advance for your input. Dina is a final year PhD candidate looking for a new research home. Please keep the podcast coming. I'm totally addicted. I have compared TWiV to others such as Cell and The Naked Scientists, which are good, but your focus, linked with the breadth and depth of knowledge that your hosts bring to the table, leads to the best storytelling around. Who knew that virology could be storytelling, right?
I'll take that.
You want to take that one?
Well, yeah, congratulations, Dina, on being almost done. And, I did a career event with high school students a few years ago where we had some scientists, and we brought in kids that really had junior high kids that had no exposure to science or any scientists or anything like that. And we got comments back at the end of the day on the forums, and one of these kids wrote, "I never had any interest in science, but one thing that struck me today is I had no idea that you could choose to live your life in a way that you would be surrounded by people like this all the time, you know?" And I think that is the best thing about being a scientist. I think scientists have joie de vivre. They love what they do and learning things and talking to people. And these kinds of things just happen naturally if you surround yourself with the right type of people. But I'll also say my secret is, you know, I teach undergrads who are addicted to these things. And, you know, I know I'm old school, but the way I get my ideas, and every single good idea I've ever had, like both of them, like they both happened when I hermetically seal myself in a room at home with no distractions and a pile of papers. I don't have any cell phone near me, anything. And I sit and I think deeply about what I'm reading, and I make connections, and every idea I've ever had has come that way. I've learned that undergrads now use these as digital leashes for their boyfriends and girlfriends, and if you don't reply back in 5 seconds, you're in trouble. So when they sit down to study, they have this thing next to them and their thoughts are all fractured. And I just want to say one thing. I don't think anything great was ever accomplished by anybody in 5-minute blocks at a time. I think you have to get the distractions out of your head a little bit and allow some time to think deeply about things. And journal clubs and places like that are great opportunities. Put that cell phone down and stop checking your email and think about the discussion and what's going on, and don't let those opportunities pass you by.
All right.
I sound like I'm 80,
Sounds
right?
great. No, that's great. I think you're right.
Well, according to Carolyn, one way to get serendipitous ideas is to get pregnant.
It's true. That is actually an excellent point.
You shouldn't be checking your cell phone while you're working on that either, I think.
All right. You want to do this more often? We can. You're good. All right. We have just a few more minutes, and one of the things we do on TWiV is to have a science pick of the week, and Rich and I will do that. So let me ask Rich, what are you picking today?
Well, I'm going to complete what is a trilogy of biographies by Walter Isaacson. I've picked before Einstein and recently Steve Jobs, and there's one more. He wrote a biography of Benjamin Franklin, An American Life. And Isaacson says, in fact, that he likes to write biographies about people who are at the interface or the overlap of the humanities and science. Okay, and you can see how Einstein and Jobs and Franklin fit into this. And so there's lots of biographies of Franklin out there, not the least of which is his autobiography. But this is a terrific book, so I recommend that.
All right. Thank you, Rich. My pick is a podcast. It's called Biochemistry Revealed. It's done by two graduate students, Pat Gordon and Ashley Bennett. It's a podcast about proteins, genes, and how your cells work. And they say this is a podcast for your parents. If you want to explain to them what DNA or protein is, this is the one to do. They have done about a dozen episodes so far: the histone code, prions, vaccines, proteins, DNA. So if you're looking to recommend someone a very basic podcast, check it out. Ashley Bennett happens to be my PhD student. So, you know, I've always been proud of teaching my students how to do good science, and now apparently they're learning how to do podcasts as well, which is great. And they actually record this in my office at Columbia using our equipment, so it sounds great. So check it out. I think they do a really good job, and we'd like to get them a little more traffic. We have a couple listener picks of the week. One is from our colleague Matt Freeman, who is at the University of Maryland, Baltimore. Big fan of TWiV, works on coronaviruses, and he sends in a story, my pick of the week, man-made jellyfish from rat cells. You have probably seen this. Scientists build live cell robot jellyfish. made of silicone and live cells that can be controlled using pulses of electricity. So check that out. Thank you for that, Matt. And then we have one from Matlock, who writes, I'm a senior internal medicine resident in Oklahoma City. Longtime listener since episode 1, way back in medical school, in fact. First-time emailer. Consider this pic. It's quite a feat of graphical design and a boon to us who need to brush up on anatomy every now and then. It certainly makes my old Netter's Atlas look very last century, and best of all, it's free. It's sort of like a digital cadaver, I suppose. It's called Biodigital Human. I haven't gotten it to work, but it is a way that you can go into a body and look at all the structures without dissecting one, which is what medical students used to do. It's a virtual 3D body that brings life to thousands of medically accurate anatomy objects and health conditions in an interactive web-based platform. So that's being recommended by an internal medicine resident. Keep up the good work. Your fantastic podcasts keep me ahead of the curve every week. So you see, we get emails from a wide range of listeners. In fact, only about 30% are scientists. The rest are all sorts of other people. And finally, you can find us if you don't know. By the way, who knows about TWiV before sitting here? Raise your hand. Almost everyone. Very good.
Let's do the converse experiment. How many are new to TWiV with this experience?
Handful. Excellent. Beautiful. You can find TWiV at twiv.tv. And if you're new to TWiV, please subscribe and leave a comment over on iTunes. It makes us visible and allows more people to subscribe. We do have a Facebook fan page. I'm sorry. It's more distraction.
I don't do Facebook. I'm so old
I know, but
school.
you probably don't like it, right?
No.
Facebook.com/This Week in Virology. I've been posting pictures all week from the meeting there, and you can see them all there. We also have a Twitter account, PROFVRR, and don't forget our iPhone case. All right. We don't charge for this. You can download the image on our website and have it made by a third-party company. It's a good way to advertise your enthusiasm for virology. And of course, if you have questions and comments, send them to TWiV at twiv.tv. And remember, our world is what it is because of science and technology. We all have to reach out and teach the public about this because they don't understand that. And that's what we do here at TWiV. I'd like to thank everyone for participating. Carolyn Coyne at the University of Pittsburgh.
Thank you very much for having me.
I appreciate your changing your lunch appointment very much.
No problem.
Sara Sawyer, University of Texas, Austin. Thank you.
My pleasure.
Maybe we'll see you in the fall, Rich and I, right?
Please.
We're trying to plan a TWiV visit to
You
your
really
place.
should. We've got some great virology at University of Texas at Austin.
And we're all going to take a field trip to Buda.
Yes. Do you know where Buda is?
I do, but why would you want to go there?
We have a fan from Buda, and we've had debates over time about how to pronounce the name and all this. It's a long story.
Okay.
Yeah, we have a fan in Buda. So we figure it's such a small town, if we have a TWiV fan there, then. Rich Condit's at the University of Florida, Gainesville. Thank you, Rich.
You're quite welcome. Always a good time.
I'm Vincent Racaniello. You can find me at virology.ws. I want to thank the ASV for having us again 3 years in a row. We really appreciate it. The president, Bert Semler, program committee. This is an activity of the Education Committee, which was chaired by Becky Dutch for the past several years, and of course our local hosts, Ann Palmenberg and Paul Friesen. If you want us back next year, tell the ASV brass, which are in a meeting right now and they can't be here, that you'd like us back again. Finally, TWiV number 200 will be up very soon. It's amazing. We'll be 4 years doing this in September, 200 episodes. We're going to have a really special episode for number 200, okay? You're going to want to check this out. I'll give you a hint. It's going to involve video and we'll be in Boston. So that'll be fun. About the second week in September. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.